So in last week's video, we were looking at prokaryotic gene regulation. So looking at how prokaryotes use operons and the systems involved with that to turn their gene expression on and to turn their gene expression off. Those were done by feedback mechanisms, right? So when something built up in the cell, the system was shut off. When something was needed, the system shut on. I'm sorry, not shut on, turned on. And you might have been wondering, do eukaryotes do anything like that? And the answer is yes. So our cells are able to turn genes on and off in response to different stimuli. If you think back to those signal transduction pathways, one of the things that we looked at was how a signal can cause transcription factors to go in the nucleus and turn genes on. That would be an example of, yes, eukaryotes do that. Something that's probably even more important than that is the fact that this goes on in multicellular organisms, and it has a really, really super important job. So thinking about where you came from, and you've all seen me draw this picture a million times. You got DNA from mom and you got DNA from dad. And you started off as a cell with 46 pieces of DNA. That cell split into two, and those exact pieces of DNA were in each cell. And that kept happening through mitosis, where we made a cute little baby you, and all of the cells in you have exactly the same DNA. So why then does a liver cell look and function like a liver cell, but a brain cell looks and functions like a brain cell? If a brain cell and a liver cell have the same DNA, how are they doing such different jobs? That's where gene expression regulation comes into play in multicellular organisms. We call that differential gene expression. And we'll come back and look at this at the end of the video, probably on Thursday. But we'll focus on how, for example, a liver cell has certain genes on and off, and a brain cell has completely different sets of genes on and off. And that's what makes them look different, and that's what makes them function differently in the organism. So keep that in mind as we go through this. Over this week, we're going to be looking at the 4 different levels of gene expression regulation. So more specifically at the molecular level, how do these genes get turned on? How do these genes get turned off? How is expression of those genes increased? How is expression of those genes decreased? So the 4 levels, today we're going to look at level 1, which is chromatin modification. Tomorrow, which is Tuesday, we're going to be looking at level 2, which we're going to call transcription initiation. On Wednesday, we're going to look at level 3, which is anything that happens after transcription, which we are going to group all that together and call it post-transcription. And that includes things on this slide, and it's also going to include all of this on this slide as well. That's all going to be level 3. We'll wrap up on Friday with a special type of gene expression regulation called microRNAs that we could probably consider level 4. So that's kind of our agenda for the week. How do multicellular eukaryotes control our gene expression? So how do we make sure genes are being expressed, or how do we shut those genes off? So the first one, like we said, that we're going to do today is level 1, which is the regulation of chromatin structure. Before we dive in, let's recap what chromatin is. So the human genome is made up of 46 pieces of DNA, but those individual pieces of DNA are way too long to fit in the nucleus. So we organize them into chromatin. The basics of chromatin is, imagine this blue piece of DNA wraps around histone proteins. And I like to think of it so it doesn't get tangled, right? So the DNA wraps around the histone proteins twice. And then this goes through further packing to organize itself into less condensed chromosome or more condensed chromosomes. There's a better picture of this. Let me flip ahead for just a second. This is what we're talking about. So the blue DNA is wrapped around histone proteins, and then that wraps itself up into chromatin. That's how our DNA is organized in our nuclei. Now, this is the first place where gene regulation can be controlled. Different chemical groups can be added either to the histone proteins, so that's going to be our first place that we see a chemical group being added. And the second place that we can see a chemical group being added is to the DNA. And based on what it is added to and what modification it is, that's going to either tighten up the chromatin or it's going to loosen the chromatin. Now, before we get into those chemical modifications, the details of it, let's just look very simply. If you are RNA polymerase and you want to find the DNA so that you can get on the DNA and move down it to do transcription, right? Taking A, making U, T to A, we make RNA. Which one would give you better access to the DNA? If the chromatin is all squished up or if the chromatin is stretched out? I think we would all say that if you are RNA polymerase, this is going to be more accessible to you, right? That is the basics of what we're talking about with chromatin modification. If the chromatin is stretched out, we consider that on because RNA polymerase can get to the DNA and do transcription. If the chromatin is squished up, we consider that to be off because the polymerase cannot get to the DNA to do transcription. Now again, how does the cell do this? It does this by adding chemical groups to either the histone proteins or the DNA. So let's take that one at a time. So within this level 1, the first type of level 1 is adding a acetyl group to the histones. Okay, wait, time out. What's a histone? The histones are the proteins, right? So those are our histones. And if you're curious about what an acetyl group looks like, imagine that this is the purple blob, so this is our protein, and a group that looks like this that's a double bond to oxygen. So this is a carbon here, if you remember. A carbon double bonded to an oxygen bonded to a carbon with 3 hydrogen. That's what an acetyl group is, if you're curious. So when these are added to the histone proteins, it loosens the chromatin structure. So that's going to be stretched out. So when our cells decide, hey, we want these genes turned on, they will go in and add acetyl groups to the histone proteins of that segment so that the chromatin uncoils and makes it accessible to the RNA polymerase. This is a temporary on switch. So when the cell doesn't need this anymore, the cell will take these acetyl groups off and the chromatin will go back to being squished. So temporary. That's our first type. The second type of chromatin modification is what we call DNA methylation. This is more permanent. And instead of adding the chemical group to the protein, we are adding the chemical group to the DNA. So a little bit different figure, but it's still looking at the same thing. So let's get our bearings here. The black string is the DNA, and the things that look like drums are the histone proteins. So this is squished, this is stretched out. A methyl group is a CH3. So every red spot right here is a CH3 added to the DNA, not the protein, but the DNA. This causes the chromatin to squish up. So methylated DNA is off. And this is actually more permanent. So this can cause long-term inactivation of genes. And there's a whole field of genetics called epigenetics that's so interesting that looks at your epigenetic modification. So they can take DNA samples and looking at how much methylation you have, how squished up your genes are, they can kind of tell you things that you've done in your life. So negative things like smoking, poor diet, not exercising, all of those poor healthy choices tend to methylate your DNA, and that can actually be a more permanent thing that's going on with your genes. What's really scary is that sometimes this can be transmitted to your children. And specifically, this can happen when you are pregnant. So, my females out there, when you are pregnant, the choices that you make and the things that you take into your body can permanently change your child's DNA as well, which is very scary. To take this even further, there's a whole study looking at babies who get cuddled more. So babies that get cuddled more have less methylation than babies that don't. So I have a really cool article for you to read, and then a discussion board type thing for you to do on a Google Doc. I tried to come up with a way to post a real discussion board on my website, but I just could not find a nice way to do it. So we're kind of going not so nice and doing a Google Doc. So every period has a Google Doc with the prompt. So read the baby's article about epigenetics and this whole crazy thing about methylation. And then respond to the prompt in the Google Doc. And then come back later and comment on at least 2 or 3 other students' things that they've come up with. Really excited for you to read this article. It's really interesting. As you know, I'm in front of my computer all day, every day. If I'm not, I have my phone with me. So if something doesn't make sense from this video, send me a quick message, send me a quick email, and I will be happy to help. Have a good week.